All right, guys, it is 10 o'clock, so we're going to get started. If someone would unmute and just let me know that you can hear me and that you can see my screen, I would appreciate it.
Yes.
All right, thank y'all. Thanks for being here today. We're going to get started with week 4, but before we do, please remember to keep your mic and camera turned off during the session. It just makes Google Meets work better. If you have a question, please unmute and ask it. We want you to ask questions if you have them. I just turned on the recording for the session, and the recorded version of this will be posted on the A+ College Ready Science YouTube channel. You can just search YouTube for that name. You do need to spell out plus. And the week 5 version of this session will be held next Thursday, the 24th at 10 o'clock. All right. Today's session is going to focus on cells. You guys know cells are the basic units of life. A lot of books call them the basic unit of structure and function in life. So they make up all living things, and they do the things that living things do. They carry out the functions. The cell is an example of what we call an emergent property. By themselves, the individual pieces of a cell, like the organelles, can't really do anything. But when you put them together, you get magic, you get life. An organism emerges, life emerges when you put all these disparate pieces together. So there's 2 main types of cells on Earth today. There's the prokaryotic ones and the eukaryotic ones. I always like to talk to my students about the word parts, the parts of a big word like prokaryotic and eukaryotic. Pro, P-R-O, means before. Karyo is used in biology to mean nucleus. It actually means like kernel or something like that, but in biology, it's used to mean nucleus. So prokaryotic cells mean before the nucleus. EU means true. So eukaryotic means true nucleus. So prokaryotic cells, the only organisms on Earth that have those today are your true bacteria, your regular bacteria, and your archaea. Used to be called archaea bacteria. We think they evolved before the evolution of the nuclear membrane, and therefore the nucleus. They have DNA. It just floats around inside the cytoplasm or cytosol. It's not enclosed in a membrane. They also don't have any of the membrane-bound organelles, for example, like lysosomes or mitochondria, chloroplast, Golgi bodies. They're missing all those membrane-bound structures. Compared to eukaryotic cells, they're pretty tiny. There's not a whole lot of diversity in terms of different types of bacteria, but there are more bacteria on Earth, obviously, than there are anything else. We think bacterial cells came first, maybe somewhere around 3.5 billion years ago, and that eukaryotic cells evolved from them. So all organisms on Earth, other than the regular bacteria, the eubacteria, and the archaea, are made of eukaryotic cells. They have a membrane-enclosed nucleus, and they also have all those membrane-bound organelles that we talked about a minute ago. So how did eukaryotic cells come from prokaryotic cells? That's a big question, and one of the main explanations is called the Endosymbiotic Hypothesis. And notice it's a hypothesis because it's really, really hard to test this. This idea was proposed by a female scientist, Lynn Margulis, in the 1960s. And she was sort of made fun of for a while at first. Most scientists didn't buy the idea. But today, it's a well-accepted idea amongst lots of scientists. What she hypothesized was that some prokaryotes began living inside other prokaryotes in symbiotic relationships. One inside the other, smaller ones living inside larger ones. And that both the symbiotes, both the partners in that relationship, gained a survival advantage. And that survival advantage was so strong that eventually this symbiotic relationship evolved into a eukaryotic cells. The smaller organisms likely gained protection. They probably were also provided with food sources by the larger organism. The larger organisms might have gained energy production. In a lot of times we think that chloroplasts and mitochondria started out as single-celled prokaryotes. So chloroplasts can do photosynthesis. Mitochondria can do cellular respiration, both functions that can produce energy molecules for organisms. We also think that things like the flagella started out as individual bacteria. So both the smaller organisms, the smaller prokaryotes and the larger prokaryotes, gain some type of advantage. And over time, these pieces switched DNA segments, and it became a permanent kind of relationship. Yes. So the smaller prokaryotes eventually became the membrane-bound organelles living inside the larger prokaryotes. And this, in turn, developed into the eukaryotic cell. So there is some, even though we can't test it, there's some evidence that backs up this hypothesis. And oftentimes we talk about this evidence in terms of the mitochondria and chloroplast. So mitochondria and chloroplast both have their own single circular chromosome that's separate from the DNA in the nucleus of a eukaryotic cell. And that single circular chromosome is shaped very similarly to the DNA found in bacteria. It's about the same size, about the same shape. Mitochondria and chloroplast also have their own ribosomes, separate from those found in the cytoplasm or on the rough ER of the cell they're found in. And these ribosomes actually are very similar in structure to those found in bacteria. So another piece of evidence. Another piece is that mitochondria and chloroplast can independently reproduce separately from when the cell they're living in reproduces. And the way they reproduce is very similar to binary fission, which is how bacteria reproduce. Also, mitochondria and chloroplast have their own double phospholipid bilayer. So they're surrounded by essentially their own cell membrane, very similar to what you would find in a bacterial cell. So this might show you that these original symbionts were phagocytized by the larger cell that they eventually became part of. And then one piece of evidence that's not listed on this PowerPoint, mitochondria and chloroplast are very similar in size and shape to modern day bacteria. So even though we can't really test the hypothesis, most scientists accept the idea that eukaryotes evolved from prokaryotes. And one of the main ways this happened is through endosymbiosis. Smaller prokaryotes living inside bigger ones, giving them an advantage, and eventually becoming an integral part of all of those eukaryotic cells. The AP exam loves to ask about the endosymbiotic hypothesis. So make sure that you know what it is, its purpose, and make sure you know some evidence to support it. Here we're just seeing how that might have happened. So here might be the first proto-eukaryote. So it's not a eukaryote yet. It's a prokaryotic cell with a piece of DNA. Eventually that gets a membrane around it to make a nucleus. And then these smaller prokaryotes like aerobic bacteria, photosynthetic bacteria start living inside and over time become an integral part of the bigger cell and form essentially organelles, eukaryotic organelles. All right, switching topics. Now we're going to talk about surface area to volume ratio. So as cells get bigger, the surface area to volume ratio for that cell goes down. So as cells get bigger, surface area to volume ratio decreases. I want you to think of the surface area like the cell membrane of the cell, the outside part, and the volume as the internal parts of the cell. So essentially what we're saying, as the cell gets bigger, the outside, the membrane, grows more slowly proportionally than the inside does. What that means, as the cell gets bigger, it gets harder and harder for a cell to transport things in and out, because one of the cell membrane's main jobs is transporting things. So it becomes very hard for big cells to transport in all the nutrients and oxygen that they need and to transport out all the waste and CO2 that they need to get out. So smaller cells have a higher surface area to volume ratio and are better at transporting things. Larger cells are not as good at transporting and have a hard time getting in enough nutrients, getting in enough oxygen, and getting rid of their waste. So that's one of the main reasons that large organisms like ourselves are made of trillions of tiny cells instead of just a few really big cells. So on your AP Biology formula sheet, there's a whole section of formulas dedicated to surface area and volume calculations. And actually, the new version of the formula sheet actually has more surface area and volume formulas than this piece does. Let's just start with a simple one, though. Let's take 3 cells, 3 cubes in this case, 1 centimeter on each side for the first one, 2 on each side for the second one, and 3 on each side for the third one. Well, you can look at your formula sheet. If you want to find the surface area of a cube, you're going to square the sides, s squared, and you're going to multiply it by 6 because there's 6 sides on a cube. So for this shape, it would be 1 squared would be s squared times 6 to get a surface area of 6 centimeters squared. For the volume of a cube, you use the formula length times width times height, which for some reason is not on this, yeah, there it is. Volume of a rectangular solid, which is a cube, length times width times height. So in this case, 1 times 1 times 1 to give us 1 cubic centimeter of volume. The surface area to volume ratio, you essentially list the surface area, put a colon, put the volume. So 6 to 1. For the 2 centimeter cell, square 2, side square to get 4, multiply that by 6 to get 24 centimeters squared of surface area. To get the volume, length times width times height, so 2 times 2 times 2 to get 8 cubic centimeters. So your surface area to volume ratio would be 24 to 8. Now, oftentimes you reduce that, just like you would reduce fractions. So you could divide both pieces of this by 8 and end up with a surface area to volume ratio of 3 to 1. For the biggest cell, the 3 centimeter cell, square 3 to get 9, multiply that by 6 to get 54 centimeters squared of surface area. For the volume, length times width times height, so 3 times 3 times 3. So you end up with a surface area to volume ratio of 54 to 27. Again, you want to reduce that like you reduce fractions. So divide both sides by 27 to end up with a surface area to volume ratio of 2 to 1. So what you're seeing as the cells get bigger, the surface area to volume ratio gets smaller from 6 to 1 to 3 to 1 to 2 to 1. So that essentially means it's harder and harder for the cell to transport things in and out as it gets bigger. Now, there's ways around this in biology. So here's some adaptations for increasing the surface area to volume ratio. One of those is just to stay small. That's probably the most important one. But other ways is to have a folded membrane, like in the mitochondria. The inner membrane of the mitochondria is all wavy. So that increases the surface area to volume ratio. Inside the chloroplast, there's all these pancake-shaped structures. That increases surface area to volume ratio. In your stomach, there's all kinds of folds, very similar to that membrane of the mitochondria. Those folds are called rugae in the stomach. In the small intestine, the inside wall of the small intestine is, again, all wavy like that. And the waves actually have waves on them. Those waves are called villi, and the waves on top of the waves are called microvilli. Those structures are all about increasing the surface area without increasing the overall volume. It's really, really important in the small intestine because one of the small intestine's main jobs is to absorb nutrients into the blood. It needs a lot of surface area to ensure that it absorbs all those available nutrients that come in through your food. Another adaptation is for long, thin cells. So long, thin cells have a much higher surface area to volume ratio than short, thick cells. And in addition to the importance of surface area to volume to transport, the surface area to volume ratio is also important for determining the rate of heat exchange between an organism and its environment. Small organisms transfer heat with the environment much more easily. Big organisms are much more efficient and keep heat in better than small ones do. All right, so let's switch gears now and let's talk a little bit about prokaryotes. Not going to go into much detail. Remember that organisms that are prokaryotic are always single-celled, always unicellular. Prokaryotic cells have a single circular chromosome, which you see right here in the diagram. It's not enclosed in a membrane, so don't let that red line make you think it's enclosed in a membrane. It's just floating, essentially. Prokaryotic cells have ribosomes. They're also freely floating in the cytoplasm. Their job is to make proteins. All cells have to have ribosomes because DNA codes for proteins, and ribosomes use that code to actually make those proteins. All cells must have ribosomes. Prokaryotes also have cytoplasm or cytosol, but they don't have any membrane-bound organelles. They don't have mitochondria, chloroplasts, lysosomes, Golgi bodies, endoplasmic reticula. Most prokaryotic cells do have a wall around the cell membrane, and you can see that here in sort of a bluish color in our diagram. This structure's job is essentially to protect the cell membrane underneath, and it also helps to protect prokaryotes that live in water, in aquatic environments, from bursting due to the water pressure building up inside. The cell wall in bacteria is usually made of a substance called peptidoglycan, which is sort of part sugar, the glyc, and part peptido, the part protein, the peptido part. Some bacteria on top of the wall have a capsule, which is this sticky mucus-like coating that helps bacteria to adhere to surfaces. It can also help to protect bacteria from immune responses. Many bacteria also have these hair-like appendages, which you see up here on our diagram. They're called pili, and these can be involved in lots of things. They can help bacteria to join together, join to each other in a process called conjugation, in which they swap genetic material. Pili can also be involved in movement and attachment to surfaces. And although we often think of bacteria as germs that make us sick, most bacteria are really, really beneficial to both us and the environment. They're involved in the decomposition of things. They help to recycle minerals through the environment. They fix nitrogen and make it available to essentially all of life. Many bacteria can do photosynthesis. They help us manufacture foods. They help us manufacture chemicals. So bacteria, there are way more good ones than there are bad ones. So eukaryotes, on the other hand, include plants, animals, fungi, and algae. We think that the first eukaryotic cells evolved somewhere about 2.7 billion years ago from prokaryotic descendants, probably through endosymbiosis. Compared to prokaryotic cells, the eukaryotic cells are a lot bigger. They're a lot more complex. Eukaryotic cells have a membrane-bound nucleus, usually with multiple chromosomes inside, what we call linear-shaped chromosomes. And they also have many membrane-bound organelles. So now let's talk about the cell membrane, also called the plasma membrane. First of all cells have a cell membrane, regardless of if they have a cell wall or not. They always have a membrane. The membrane's job is to allow cells to establish and maintain an internal environment. So essentially to separate themselves from what they're surrounded by. That membrane also allows the cells to transport materials in and out. It may be involved in the immune response, has all kinds of functions. But essentially, this bold statement is what the membrane mostly does. Establish and maintain an internal environment different from what the external environment is. The membrane is selectively permeable, or some people say semi-permeable. This means it allows some things in while keeping other things out. Typically, small and non-polar substances can go through the cell pretty easily, and larger, more charged, more polar things have a much harder time getting in. The main component of the cell membrane in all of life is the phospholipid. We're going to look at a picture of phospholipids in a minute, but it's important to know that phospholipids are considered to be amphipathic molecules. And this means they have part of the molecule that's polar, charged, and another part that's nonpolar. And here we can see that. So a phospholipid is made of a phosphate group, which is polar. It's what this red part represents. And two chains of carbons and hydrogens called fatty acids. Everywhere there's a bend in these little wavy structures, there's implied to be a carbon atom with hydrogens attached to it. So these are long carbon hydrogen chains. These guys are non-polar because there aren't really any highly electronegative atoms in them. And this part is highly polar because of all these electronegative oxygen atoms. So when a bunch of phospholipids are put into water, the phosphate groups, the polar heads, are attracted to water, and the non-polar tails are repelled by the water. They're hydrophobic, whereas the fatty acid head, the phosphate heads are hydrophilic. They like water, are attracted to water. So what that ends up causing is it causes the phospholipids in water to form themselves into two-layered structures, what we call a bilayer, with the polar heads, the phosphate heads, pointing outside where there's water and also pointing inside the cell where there's water. And the fatty acid tails oriented toward each other away from the water. So cell membranes mostly are composed of a bilayer of phospholipids. It looks very much like that. Now, there's more to it than just the phospholipids. Our current understanding of the cell membrane is called the fluid mosaic model. Fluid, because these phospholipids are very much like a vegetable oil. They're sort of liquidy. And mosaic, because there's all kinds of things embedded within those phospholipids. Everything you see in this picture that's blue is a protein. So lots of proteins embedded. Those proteins give the cell membrane lots of functions. We'll talk about some of those in a minute. And the green structures are sugars, carbohydrates. And then the bright yellow thing you see there is cholesterol. Cholesterol helps to make the cell membrane a little more solid, helps to keep it from freezing. And there's cholesterol molecules embedded throughout the cell membrane. Let's talk about just a few of these proteins. So some of the proteins are called integral proteins because they go all the way through the cell membrane. And oftentimes these kinds of proteins act as channels. They allow certain very specific substances to cross either into or out of the cell. There are other proteins that may be on the outside of a cell, like these guys, that can act as what we call receptor proteins. So these can receive signals from cell signaling molecules like hormones or other kinds of cell signals like growth factors, and essentially then send that message into the cell. Other proteins, like the glycoproteins that are combined with sugars, may act to identify a cell. They may act sort of like name tags for the cell and essentially tell the organism's immune system, hey, this cell belongs to me. This is me. Please don't destroy it. These glycoproteins can also be involved in cell signaling. So there's lots of functions that can happen due to the proteins embedded in the more liquidy phospholipid bilayer. We'll talk a lot more about these proteins in our week 5 session. That session is going to be devoted to transporting things in and out of a cell. So let's talk a little bit more about eukaryotic cell structures. So we've already mentioned the membrane, holds the cell together, helps to regulate what can come in, what can exit, helps to maintain a constant internal environment. The nucleus contains the DNA, controls the activities of the cell by telling the cell, make these proteins, here's how to make them. Those proteins then go on to determine the traits of the organism and cause the reactions that happen in the organism to happen. The cytoplasm or cytosol is just the fluid-filled space that contains the organelles, takes up most of the volume. And then the organelles are the specialized structures that each one has a specific function. By separating pieces of the cell into compartments, which is what the organelles do, it allows these spaces, these compartments, to specialize. That allows the different parts of the cells to divide up the labor and make the cell much more efficient. It's very much like an assembly line. One person could build a car, but probably not very well. But if you give each person in an assembly line one job, those individuals become really good at that job, and the car can be built very quickly. That's what organelles do for a cell. Allow certain pieces to specialize, divide up the work, making the cell much more efficient. Notice the bold statement here. It's important to note that the number and distribution of organelles is very different from cell type to cell type because the cells specialize. A cell that uses a lot of energy needs a lot of mitochondria. A cell that makes a lot of proteins and transports those proteins out of the cell may need a lot of rough ER. We'll talk more about those structures, those organelles, as we go. So we've already talked about the nucleus. It's the control center for the cell's activities because it's where the DNA is at. It's surrounded by a nuclear envelope, which is actually a double phospholipid bilayer. So it's really 4 layers of phospholipids. It has pores, which allow certain things to go in and out of the nucleus, things like the messenger RNA, which needs to leave the nucleus and go out to the ribosomes. The ribosomes are cell particles. They're not enclosed by a membrane. They're made mostly of a type of RNA called rRNA with some proteins. They're not usually considered organelles because they're not enclosed by a membrane. All cell types have them. Their job is to make proteins. They're the sites of protein synthesis. And their presence in all cells is an example of common ancestry of all living things. All living things on Earth, at least, have ribosomes. And this is evidence that all life on Earth came from an original living thing. There's 2 types of organisms, or 2 types of ribosomes, rather, that exist, and we base those differences on location. There are free ribosomes that essentially are freely floating in the cytoplasm of the cell. They're found in all types of cells, prokaryotes, eukaryotes, all examples of those. These make proteins that essentially stay in the cell that made them and function within that cell. And then there are bound ribosomes. These are typically attached to the rough ER, the rough endoplasmic reticulum. They're found only in eukaryotes. Prokaryotes don't have a rough ER. And these make proteins that are going to be packaged and shipped. And those proteins are either going to be shipped out of the cell to other parts of the body, or those proteins may end up in the cell membrane where they'll function. So bound ribosomes make proteins that are either shipped out or end up in the cell membrane, or in some cases, the membrane of the lysosomes. Many of these proteins are cell signaling molecules, or they may also act as antibodies to help us fight off infections that are found all throughout our body. So I already talked a little bit about compartmentalization, but it's a really important theme. The membranes are the internal components, the internal parts of a cell, at least in eukaryotes, are divided up into specialized areas, organelles. And this allows those areas to take on very specific functions, become really efficient at those functions, and divide up the labor so that the overall cell becomes much more efficient, much better at carrying out complex processes. So let's talk about the endoplasmic reticulum, the ER. It's essentially composed of a network of small membranous tubes. They're called cisternae. I don't really think you need to know that term, but know they're made of small membranous tubes. It's always found just outside and around the nucleus. There's 2 types in eukaryotes. There's the smooth. It's smooth because it doesn't have ribosomes on its surface. This structure helps to make lipids, phospholipids, and steroids that are important in the cell. It also helps in the breakdown of sugars, carbohydrates. The smooth ER helps to detoxify the blood. So there's lots of smooth ER found in liver cells. It also helps to store calcium ions, which are really important for muscle contractions. So muscle cells have lots of smooth ER. The rough ER is the one we talk more about in biology classes. This one is covered with ribosomes. That's what makes it appear to be rough. It's involved in protein synthesis, protein packaging and modification, and protein transport. So the rough ER packages the proteins made by the ribosomes and then ships those out. Before those proteins leave the cell, though, they go to our next organelle on the list here, which is the Golgi apparatus. You also might hear this structure called the Golgi body or the Golgi complex. So the proteins from the ribosomes and Receptor go here. They're modified in the Golgi body. Some things might be attached to them, like sugars or lipids. And this helps them to function. The Golgi can also act as a warehouse and store those proteins until they're needed. But eventually it packages them in vesicles, membrane enclosures, and ships them out toward the cell membrane. Lysosomes are another membrane-bound organelle found in eukaryotes. And these contain really powerful enzymes and also acids. Sometimes we refer to these enzymes as hydrolytic. That means they hydrolyze things, they break down things. So breakdown enzymes. They help to carry out the process of what's called intracellular digestion within a cell. That means inside the cell. So things the cell phagocytizes, takes in, are then broken down with the help of the lysosome. Once broken down, the components of these materials then can be reused within the cell. It's also been found that lysosomes help to break down old cells through a process called apoptosis, or programmed cell death. We also need to mention vacuoles or vesicles. These are essentially membranous structures that store materials within a cell. They can store food, they can store water, they can store waste, and there's actually a specific type of vacuole called the contractile vacuole, which takes in water and helps to pump it out of freshwater organisms so they don't build up too much water inside. These are found in organisms like protists, like euglenas and amoebas and other freshwater protists. So let's next talk about the endomembrane system. So this is a group of membranes and organelles only found in eukaryotes that together work to modify, package, and transport lipids and proteins throughout and even out of a cell. So once bound ribosomes make proteins, those proteins enter the Receptor. Here they're packaged, maybe modified a little bit, and they're surrounded by a phospholipid-based membrane that forms what we call a secretory vesicle. These vesicles break away from the Receptor and move toward the Golgi apparatus. Inside the Golgi apparatus, the proteins may get lipids or sugars added to them, so they're modified. Once that modification is done, they break off again in a lipid-based vesicle and move toward the cell membrane. So essentially, the general pathway is the proteins move from the Receptor into a secretory vesicle to the Golgi apparatus. There they're modified. They break away again in a secretory vesicle, which will then head for the cell membrane where the proteins will be released. And again, remember, sometimes those proteins become part of the lysosome or the cell membrane. Here we can see the endomembrane system happening. So here's the rough ER. Proteins are made, put in a transport or secretory vesicle, fuse with the Golgi apparatus. There, sugars or lipids are added. They move through that Golgi apparatus and eventually break away in another secretory vesicle, bump into and become part of the cell membrane. The phospholipids in the membrane become part of the cell membrane, help it to grow. And then the proteins that were inside that vesicle are either pushed out or become integrated into the cell membrane. So that's called the endomembrane system. Its job is essentially to make and transport proteins throughout the cell and eventually out of the cell. All right, so the last couple of cell structures we're going to talk about, number one, are the mitochondria. You guys know from ninth grade biology and even middle school, they're oftentimes called the powerhouse of the cell. They don't make energy. They make ATP, which is the body's energy storage molecule. But they have to get the energy from somewhere. The energy to get that to make that ATP comes from food. So the mitochondria take energy from food and transfer it to a molecule called ADP. I want you to think of ADP like a dead battery. So they charge it up with energy from food, add on another phosphate group to make adenosine triphosphate, ATP. That molecule, I want you to think of it as a charged battery. Almost every reaction in your body runs off energy from ATP. So ATP's job is to store and transfer energy and make it available for the body's processes that need it. The mitochondria has its own DNA. We talked about a minute ago how that it's a single circular chromosome, very much like the DNA found in bacteria. It has its own ribosomes, again, very much like bacterial ribosomes, has its own enzymes, and remember that it can reproduce independently of the cell that it's living in by a process that looks very similar to binary fission. The evolutionary significance is that we believe that mitochondria started out as aerobic bacteria capable of doing aerobic cellular respiration, and that eventually some of those began to live in symbiotic relationships with larger prokaryotes. The small aerobic bacteria gained the advantage of protection and probably food sources, and the bigger organisms gained a part that could make ATP for it. So it was an evolutionary advantage for both, and that this ultimately became a really important symbiotic relationship and ultimately gave rise to eukaryotic cells. Let's also talk for a minute about chloroplasts, which are found in plants and the green algae, or algae in general. Chloroplasts are a type of plastid or pigment container. Like the mitochondria, they have their own single circular piece of DNA, their own bacteria-like ribosomes, and their own enzymes. Also, like mitochondria, they can reproduce independently of the cell they're found in through a process that looks very much like binary fission. So this too indicates that they may have evolved from some type of blue-green bacteria, cyanobacteria, that formed a symbiotic relationship with a larger bacteria. So the chloroplast is made of these structures inside called thylakoids. These are little sacs of chlorophyll containing bags. I want you to think of chlorophyll almost like a solar panel. Chlorophyll's job is to absorb solar energy and make it available to other parts of the cell. So the thylakoids contain the chlorophyll. These thylakoids are stacked within a cell to increase the surface area found within. And they're surrounded by this cytoplasm-like fluid called stroma. The stroma is the part of the cell where the second part of photosynthesis, the Calvin cycle, happens. So part one of photosynthesis happens on the thylakoids. That's when solar energy is absorbed, and that energy is used to make ATP and another energy storage molecule called NADPH. Part two happens in the Calvin cycle, the liquidy part of the chloroplast, and that's when the energy from these two molecules is transferred to carbon-carrying molecules to make glucose or sugar. So the Calvin cycle is when the sugar is actually made. It's part two of photosynthesis. All right, cells also have an internal support structure called a cytoskeleton. This structure is usually made out of proteins. It helps, again, to support and protect the inner workings of a cell. It also helps to keep the inner organelles organized. The spindle fibers that you guys have heard about when you've talked about mitosis or meiosis are made of parts of the cytoskeleton. So these parts also help to make up the flagella and the cilia. So the cytoskeleton is involved in movement, whether we're talking about movement of materials within the cell or movement of the cell itself. Motility is another word for movement. Again, the cytoskeleton is made of various size pieces of proteins. You might hear those called microtubules, microfilaments, or either intermediate filaments. But they're just pieces of protein. Some cells have cell walls. Remember that animal cells do not have them. Plant cells do. Fungal cells do. And bacterial cells do. In plant cells, that wall is outside of the cell membrane, It's made mostly of this complex carbohydrate called cellulose, which is very hard to digest, but also very hard to break down. It's very strong. Its job is to support the cells, because remember, plants don't have a skeleton like you do. They don't have bones. So it's to provide support. It's also to protect the plant from bursting when the plant cell takes in too much water. Fungal cells have similar functions, but they're made of a completely different carbohydrate called chitin. But again, the function of the cell wall is to provide support and to protect from bursting when the cell's in an aquatic environment. Bacterial cells are also mostly made of complex carbohydrates, very oftentimes a material called peptidoglycan. So we're going to end this session today by looking at an FRQ from an AP exam from 2 or 3 years ago. So you guys just take about 2 minutes and read through all 3 parts of this question, A, B, C, and D, and go ahead in your mind and try to formulate an answer. I'll be back in like a minute and a half or so, and we'll talk about what the answers to these parts are. All right. So first of all, if you read the beginning of this question, it might have been intimidating. You probably never heard of a CFTR protein. You may not have even never heard of a gated ion channel. And it turns out to answer this question, you don't really need to know what any of those things are. And that's true in a lot of AP questions. Let's break it down and look at the parts. Part A, in the provided model of a cell, draw arrows to describe the pathway for production of a normal CFTR protein from gene expression to final cellular location. All right, so it's important to remember that the CFTR protein is found in the cell membrane. That's where the protein is at. But we want to know, how does it get there, essentially, is what part A is asking us. Well, you guys know that DNA in the nucleus tells the cells how to make the proteins. And that DNA first has to be transcribed into RNA, which is going to leave the nucleus and move to the ribosomes. So let's draw an arrow starting in the nucleus and head it out. And that mRNA is going to go, in this case, to ribosomes on the rough ER. And we know it's on the rough ER because the CFTR protein ends up in the cell membrane. Remember that proteins that are shipped out of a cell or proteins that end up in the membrane are made by the ribosomes found on the rough ER. Proteins that just function within the cell are made by these freely floating ribosomes, like the ones I'm circling there. So our first arrow is going to be from nucleus to bound ribosomes. So we know that there, we're starting the endomembrane system. The proteins are made. They go down into the rough ER, where they're modified and packaged, and eventually break off in a secretory vesicle, a little phospholipid bag, essentially, and move to the Golgi apparatus. So we're next going to draw an arrow like so, and I'll probably label mine 1, 2. In the Golgi apparatus, remember the proteins are further modified. They get sugars and lipids added to them. They also get stored until the cell needs them. Eventually, they break off in another secretory vesicle, the proteins do, and combine, fuse to the plasma or cell membrane. And that's where they end up in this case. So for part A, we just answered part A. You're going to draw those arrows on the picture on the exam, and I would go ahead and number them so that they realize what order this is happening in. 1, 2, 3. That's it. You get full credit for part A if you essentially understood that proteins that end up in the cell membrane were made by ribosomes bound to the rough ER, which sent them to the Golgi apparatus, which sent them to the plasma membrane. Part B, identify the most likely cellular location of the ribosomes that synthesize CFTR. Like we said, proteins that end up in the cell membrane are almost always made by ribosomes bound to the rough ER. So all you need to say here is that these ribosomes are probably found bound to the rough ER. Full credit if you said that on part B. And then part C, identify the most likely cellular location of a mutant CFTR protein that has an amino acid substitution in the ATP binding site. Well, this question told you that these ion channels help things to move across the membrane. So whether they're working or not, those proteins have to be found in the membrane because it's their job to move things across. So essentially all you need to say on part C is that these proteins would be found embedded in the cell membrane, or just cell membrane would probably be good enough. Cell membrane or plasma membrane. All right, so this was a fairly easy FRQ, and really all you needed to know to answer this question is you had to know how the endomembrane system works. Remember, the endomembrane system is the system of organelles that takes DNA, makes a protein, packages that protein and modifies it, and ultimately ships it either out of the cell or helps it to become integrated into the cell membrane, which is what happened in this case. All right, that's it for our session today. Let me stop and answer any questions that you guys might have. Anyone have questions that I can answer for you about Unit 2? All right, well, thanks for joining us today. I hope you guys check back in with us next week, same time, Thursday at 10 a.m. And we'll be talking about how things move into and out of a cell. And we're also going to do a little bit of biology math on a topic called water potential, which is confusing to a lot of students. So next week would be a really good session for you to attend. All right, hope to see you there.